1, 2, 3, 4. Okay, let's talk a little bit about the types of fast synapse signals that are going to be picked up by this postsynaptic cell. And most of these, from fast synapses, most of these signals are going to actually be graded potentials on the membrane of this postsynaptic cell. And there's two specific kinds of graded potentials that we're going to be talking about. And the first one is an excitatory postsynaptic potential, or just EPSP for short. And these are going to be postsynaptic potentials that are depolarizations, or changing the polarization to bring it closer to threshold potential for an action potential. So that's why they're called excitatory, because they're taking this membrane closer to action potential, or threshold for an action potential. Just as this is an interesting point here, one of the most common neurotransmitters that's going to be used in excitatory synapses is glutamate. This is just the same alpha amino acid that is being used in the proteins and other things in your body like that. And so that's kind of an aside there. Also, most of these channels, these ligand-gated channels, are going to be picking up in these excitatory postsynaptic potentials are cation channels. And so these are the general cation channels that are going to be letting through most or the common positive ions that are in the area. So this is going to be sodium and potassium. Now remember, the potassium leak channels are open, and so potassium is going down its concentration gradient quite a bit compared to sodium. So when these cation channels open, they're going to predominantly let through sodium, because sodium has more of a electrical and chemical gradient that's going down. And so the net effect of just these general cation channels opening is going to be towards depolarization, because far more sodium is going to be going through these channels. The other type of postsynaptic potential is going to be inhibitory postsynaptic potential. And this is going to cause hyperpolarization, or taking the potential further away from threshold potential, and so it's less likely to be able to reach threshold. And the most common neurotransmitter that's used in these inhibitory synapses is going to be gamma-aminobutyric acid, or just GABA for short. And most of the channels that are going to be used here are either chloride channels, so chloride being a negative ion and is going to be rushing out of into the cell from the extracellular fluid, rushing into the cell, causing it to become more negative and hyperpolarizing the cell. The other one is specifically for potassium ions, another potassium ion channel. And again, as we let the potassium ions back out of the cell, taking their positive charges with them, that's going to cause hyperpolarization in these cells. So those are the two major kinds of these postsynaptic graded potentials that we're going to be having in these fast synapses. Let's just take a moment to review or talk a little bit about these slow synapses as opposed to the fast synapses. I'm not going to be talking a lot about them, but just a little bit. And really, they have the same basic release mechanism for their neurotransmitters as fast synapses do. You're going to be having the action potential coming down to the axon terminals. It's going to open the voltage-gated calcium channels, letting the calcium in, and this is going to cause vesicles to bind to the bilipid membrane, releasing neurotransmitters. One of the big differences, there's a couple different differences here. The major difference is slow synapses are going to generally have neurotransmitters that are activating a second messenger system in the postsynaptic cell rather than just an ion channel. And so the second messenger system, because there's a lot more going on there, these are going to be a slower action. But the other part about this is that this is also going to tend to be a long-acting effect as well. One of the common types of neurotransmitters that's being used here are neuropeptides. Neuropeptides are a lot larger than the classical neurotransmitters like dopamine and serotonin and glutamate. These are often going to be strings of amino acids together, somewhere between 2 and 40 amino acids all chained together, so they're a lot larger as well. And a lot of the other things that are going to be used here are what are known as neuromodulators. So a lot of these neuropeptides are actually neuromodulators. And neuromodulators are not going to open ion channels are not going to cause a postsynaptic potential. What they do is they'll act in some other way, generally through these second messengers or something like that, that is going to depress or perhaps enhance synapse activity in the future. So it can make this synapse more sensitive or less sensitive as time goes on. And one interesting note about neuromodulators is that these can also be released from fast synapses alongside classic neurotransmitters as well. So sometimes these are going to be used in those fast synapses as well. Okay, so enough of the different kinds of synapses. We're going to really move on to now is kind of summing this all together and start talking about signal integration in these neurons. And what's going to happen is as we begin plowing all of these graded potentials into the dendrites and the cell bodies of these postsynaptic neurons, these graded potentials are going to begin to merge and create what's called the grand postsynaptic potential. The grand postsynaptic potential is just the summation of all these different graded potentials that were caused by these both other neurons and these synapses, but also sometimes these graded potentials can be coming from some other sort of activation of that neuron as well. So we'll have both of those sort of things going on. And this summation can either be temporal or it can be spatial. So we can be summing in time, and we can also be summing in space. That might be a little bit confusing, but let's talk a little bit more about how that could happen. Okay, we have a model of what's going on with all this stuff here. And right through here, this is a cell body of some neuron. We have a couple dendrites that are moving off. We have the axon hillock right here, and then some myelin that's taking on the axons that moves away from the cell body. And this big gray pointy thing here is just some imaginary electrode that we stuck into the axon hillock so we can measure in this cartoon the potential that's happening right here. Up here in the upper left-hand corner is this single, now again this is a cartoon, oftentimes cell bodies or neurons are going to have thousands to hundreds of thousands of synapses on, but we're just going to be looking for the moment at this single one synapse right here. And so we have this one axon terminal synapsing onto this. Okay, so let's take a look at what could potentially happen here. Is that when this sucker fires, oftentimes, or essentially never, is a neuron-to-neuron synapse going to cause enough depolarization to actually bring this to threshold. So you look down here in our axon hillock, if that fires just once, we get a little bump, a little graded potential, but it's not even close enough to get it to threshold. However, if it starts firing repeatedly over and over and over, you can get something that looks like this. Now go ahead and look down at our membrane potential in our axon hillock here down in the lower right hand corner. Now we have a bump, but then we have a second bump, and a third bump, and a fourth bump, and a fifth bump. So what we're getting is these graded potentials that before the first one dies off, we're getting another one, and another one, and another one, and spatially, excuse me, temporally. So in time, they're summing together and able to get this to threshold and cause an action potential to go down this axon by summing these graded potentials spatially, temporally. So summing them together in time. Well, that's not the only way that we can be summing together these graded potentials. Imagine we have now another synapse on here, so now we have two, and imagine these both have essentially the same type of small graded potential that we saw before. Each one individually can cause a graded potential that's going to get it part way to threshold, but not all the way there. But these graded potentials, as they move across the cell body and towards the axon hillock, they're going to sum together. And what you would actually get at the axon hillock here is one larger graded potential that will bring it to threshold, and we will get an action potential. So this is a way that we can get both spatial and temporal summation of these graded potentials, these signals coming in from other neurons, and integrate together into one signal. But that's not the only thing that can happen. These are both excitatory postsynaptic potentials. We also have these inhibitory postsynaptic potentials. So now this bottom synapse here is going to give this inhibitory graded potential. And so what you're seeing here on the lower left-hand corner is just a trace for that. And in the upper area, we're seeing a trace for an excitatory postsynaptic potential. And as these sum together, as they reach the axon hillock, you get essentially nothing. They cancel each other out. And so you get these competing signals that can essentially lead to no membrane potential by the time you reach the axon hillock. Okay, well, that's not the only way that we can get inhibition. See, this kind of inhibition is going to inhibit all of the different axons that are in that area. We can get this other kind of inhibition. This is called presynaptic inhibition. So what we have here from the upper right-hand corner is our old friend, this synapse up here that's been here all the time. But now we have, right off to the left-hand side of that axon terminal is another synapse right there. And what this can do is cause another graded potential that will affect the action potentials that are coming down towards this axon terminal here. And so one example of what could happen here is that if you look at this lower trace here, we could have this inhibitory signal, this inhibitory postsynaptic potential in this other axon right here, which would lead to essentially no membrane potential in the postsynaptic area of this cell body here, because the signal that was coming down, that action potential that was coming down this axon, has been blunted by this inhibitory signal from this presynaptic synapse that's inhibiting it. Now, these presynaptic synapses or presynaptic regulation can also be excitatory. They could also cause an increase or have an excitatory postsynaptic signal as well that would increase the probability of releasing neurotransmitter and causing postsynaptic potential in the main cell here. So those can go either way. So that's really what's happening with summation. We're getting this summing together of signal both in time and in space in both cell bodies, but then also we get these sometimes presynaptic interference or enhancement of those signals as well. Okay, so there's a little bit more to talk about here, but really I'm going to let you go and not make this video too awfully long. I want you to do just a minor amount of reading in your book before you come into class on Friday, and really it's going to add up to a total of like 4 pages. So in your book, go to section 4.6, the neuromuscular synapses. I want you to read about those. Those are a very specific type of synapse that we haven't really talked about in this video. Those are on pages 134 to 136. That'll be pretty quick. But those are going to be important, especially in lab, because we're going to do a lab later on that's very specifically looking at neuromuscular junctions. And the other thing I want you to read is a section called The Importance of the Postsynaptic Neuronal Integration. This is on page 138. It doesn't even take up the entire page. So it's a pretty minor amount of reading there. So if you can go ahead, read those before you come into class next time, I greatly appreciate that, and have a good day.